Hello,
Okay, it
hello
works.
everybody.
Hello, welcome back to Getting That Medical Degree, where we just came from watching Doctor Strange. And don't worry, no spoilers. But sadly, we're not going to be talking about that. We're going to be talking about MCAT Academy, the last episode
Yeah, so this is the last
called
episode of
Bio.
Biology, not last episode of Mcat Academy.
Woo!
So
Last episode of Bio.
I know it's been a journey. Lots of videos. This is genetics and evolution. There's a prefaces. I took like a world and it was 20 questions of genetics. I missed all 20 of them, so it's kind of why I'm reviewing this with you guys today. Here we go. So we have definitions, which is pretty important. Alleles, these are like your alternate forms of a gene. So there's a thing called a dominant allele. These are usually indicated by like a big letter. So a dominant allele requires one copy to be expressed. So it could be a big letter and a small letter, and it's still called dominant. A recessive allele requires two copies to be expressed. So you might see like two small letters. That'd be a recessive allele. But if it was a big B, like a big B and a small b, it's still counted as dominant. A genotype is basically a combination of alleles one has at a given locus. Basically, homozygous is having two of the same allele, heterozygous having two different types of allele. Phenotype is your appearance, basically. The observable manifestation of a genotype, the definition of it, but it's basically how the person looks or what happens, like what appearance this phenotype gives to something. Dominance, so it's going to be complete or codominant. So complete is only one dominant allele. So I don't know, a flower is white. That is its dominant allele for some reason. Codominance, you have more than one dominant allele. So you might have like a dominant allele that's like white and purple or something like that, and then it's like a combination of the two, like somewhat white, somewhat purple. I don't know. Incomplete, so there's no dominant alleles and it's heterozygous. So more definitions. So penetrance is basically how many people are carrying a particular allele to that also express an associated phenotype. So how many people are carrying this allele, and you go to expressivity, which is basically okay. They have this genotype, phenotypic outcomes, but what are like the different outcomes from this genotype? Like how much is it expressed, I guess. And some important information here is genetic leakage, drift, and founder effect. So leakage is like the flow of genes between species via like hybrid offsprings. Genetic drift is like when the gene pool changes as a result of chance. So founder effect and genetic drift are basically the same thing, but genetic drift founder effect is kind of like a more extreme version of genetic drift. So an example of genetic drift could be like there was a huge hurricane that hit this part of a town or something. And there's a lot of animals there, and a lot of animals died. So that was a part of a chance. So that hurricane was a chance to happen because of Mother Nature, and that changed the gene pool because changed drastically because there's barely any survivors. So founder effects basically when a bottleneck that suddenly happens, it isolates a small population, and then they start interbreeding pretty much. There's no one else. Then like the rank of like the taxonomic rank. So it goes from a kingdom, phylum, a class, a order, a family, a genus, and a species. So species being more specific to more broad. So this is like from more broad to very specific. So King Philip came over from Great Spain. It's kind of easy way to remember it. Still takes a little bit of brain power to know it, but still it's a little tricky to do. Then genetics, you have a couple of laws, Mendel's laws. So you have law of segregation. So an organism has two alleles for each gene, which segregate during interphase one. Because of this, gametes only carry only one allele for a trait. Law of independent assortment. The inheritance of one allele does not influence the probability of inheriting a given allele for a different trait, except when they're linked in genes. So in genetics, a couple of important experiments. So Griffith, so he demonstrated transformation. So a heat killed smooth strain of bacteria, still transformed rough strain to smooth. Avery, McAuley McCarthy, degradation of DNA led to a cessation of bacterial transformation, but the proteins not like degradation of proteins did not like stop it. So Hershey Chase, they confirmed that DNA is a genetic material because only radio-labeled DNA can be found in the bacteriophage that infects bacteria. Then you have some mutations. So you have different types of mutations called nucleotide mutations, and the next slide would be chromosomal mutations. So nucleotides, you have point mutations, so this is changing one nucleotide for another. And you have frame shift mutations, which are like moving the three-letter reading frame. So you have a silent mutation, which has no effect on the protein. A missense, which you replace one amino acid with another. And a nonsense, so a stop codon replaces the amino acid. These kind of make sense though. Missense, you're replacing something because it makes it not doesn't make not sense. And then nonsense, you're basically telling the body to stop. Put a stop code on randomly an amino acid. Like, oh, you're making this thing, then just stop randomly. Insertion and deletion, shifting the reading frame, which leads to a change in all the downstream amino acids. Chromosomal mutations are usually larger because of the chromosome itself. So a deletion of a chromosome leads to a large segment of DNA being lost. Duplication, a segment of DNA is copied multiple times. Inversion, the DNA is reversed. Insertion, it's removed from one chromosome to another. And translocation is like swapping DNA with another chromosome. So chromosomal mutations are going to have a lot more effect than like a nucleotide. Some techniques, so the Punnett squared, a monohybrid cross counts for one gene, a dihybrid counts for two genes, sex-linked cross is linked to the X chromosome, recombination. Frequency is basically how likely two alleles being separated during crossing over. And meiosis, so farther is more likely. And the Hardy-Weinberg principle, basically if a population meets certain criteria, then the allele frequencies will remain constant. So P, that's dominant, plus Q equals 1P. So it's like P2 plus 2P, big P little q plus Q2 equals 1Q. So, yeah. Then you have evolution. So, natural selection is basically is the main model for evolution. Modern synthesis model is neo-Darwinism, so mutation and recombination are the mechanism of variation. Inclusive fitness. Then if a population meets certain criteria, then that allele frequency will remain constant. Kind of like survival of fitness. Punctuated equilibrium, kind of a slow process with random bursts of like rapid bursts of evolution activity. And then, yeah. And then you have isolation, so reproductively isolated from each other. And then the molecular clock model, which is the degree of difference in the genome between two species. That's related to the amount of time since both species broke off from the common ancestor. Law of segregation, basically an organism has two alleles for each gene, which segregate during anaphase 1. This is why it carries only one allele for that trait. Genetic drift, what is it? So this is basically when something's killed off by chance. Maybe an earthquake happened, or a tsunami that killed and changed the gene pool. Yeah, that's it for this chapter, guys. I hope you guys learned something. This is Biology Complete now for the Mcat Academy. Any closing thoughts, Mitchell?
Thank you for tuning in.
And we'll see you next time. Peace.